Okay, so the next session, is going to be on opportunities for students in the IPN. And we have 3 people that we've invited to present. The first is going to be a presentation from Noha Gerges, who is responsible for MyTax here, and she'll talk about, I think, internship opportunities, opportunities to explore beyond the regular graduate training. We'll then hear from Crystal Van Hoof about HBHL. And finally, we'll hear from our recently launched EDI committees that have been running for the past year from Roberta LaPiana. So, Noha. Whoops. I cannot operate these things somehow. It's my... All right, there we go.
Thank you.
Thanks.
All right. Hi, everyone. First of all, thank you so much for having me here today. It's always a pleasure to come chat with the IPN group. We used to do this before the pandemic, so this is the first time that we're gathering after, so welcome. So today I'm just going to take a couple minutes to walk you through who we are, what we do, and how we can help support you while you're here at McGill. I know that some of you are already aware of MyTax, so for those of you who do know about us, please bear with me while I introduce who we are. And then for those of you who don't, buckle up. So my name is Noha, and I'm your representative from MyTax. So who is MyTax? What do we do? We are a Canada-wide non-for-profit organization funded by both the federal and the provincial governments. And our mandate is really to boost innovation in Canada. So short and sweet, that's what it is. How do we do that? So essentially, we have a lot of different funding opportunities available to you. And the reason for that is that one of our goals is really to help you build your CVs. Okay, short and sweet, that's what it is. We want to be able to help you build skills that you will need to take your next step, whatever your next step is going to be. So whether that's taking on another degree, whether that's taking a job in the academic sector, the public sector, the private sector, etc. Okay, so we have a lot of different opportunities for you to build those skills. Now, our funding opportunities are always collaborative in nature. So when you think of MyTax, you always have to think of the word collaboration. And so the idea here is that it's yourselves, your professors, and an external partner working together to solve some kind of challenge that you're interested in solving. And we'll talk about that a little bit more later. So if you know us, or if you don't, you'll know, and if you go on our website, you'll see we have tons of different funding opportunities. And so today, there's no time to talk about them all. But I have picked 2 that I think will be the most interesting to you. The first one is the Accelerate program. So what is Accelerate? Accelerate is our flagship program. It's the one that you will very most likely use if you wanted to approach us for funding. And it's really meant to support applied research projects between yourselves, your professor, and an external eligible partner. And we'll talk about eligible partners in a second, but this could be anything like a company, an eligible nonprofit, or some others that we'll touch on. Now, this program is open to academic institutions across the country, and the reason I mention that is because some of you come from labs that tend to submit team grants with professors from other universities, from other colleges, and that's totally fine with MyTax as well. So when you're thinking of MyTax, you can build a small project that will just fund yourself for a certain amount of time, or you can build projects with your professors that will support multiple trainees, multiple other professors' trainees as well, so you can really make these as big as they need to be. Now, who's eligible? A lot of people tend to think it's just masters, it's just postdocs. It's not that. It's basically everybody's eligible. So college students, undergrads, masters, PhDs, postdocs, most recently recent grads as well. So it's really open to whoever's interested in doing this. And the projects start at 4 to 6 months. So your shortest project is 4 months, and your biggest project is your entire degree. And we'll talk about those maxes in a second. Now, MyTax, contrary to what some people think, is not just STEM focused. We're open to all the disciplines. So it doesn't matter what it is you're studying. What matters is the strength of the collaboration. So that's really the most important thing that we're looking at. Is this a true collaboration? Is everybody going to benefit from it? And everybody being yourselves, your profs, and the partner. It needs to be win-win-win for everybody across the board. Accelerate is not merit-based, so we're not really looking at your publications or any of that. We're really looking at the collaboration itself. And all the sectors are of course, welcome. Now, the cool thing about Accelerate is that there's no deadline, and it's a 95% success rate. So you don't tend to hear that success rate often. A lot of people usually ask if there's a catch. There is no catch. The thing with the Accelerate program is that you're collaborating with a partner, and it is on you to find that partner, and that's the hard part. So you really need to go out there, figure out what it is that you want to do, who you want to do it with, approach that partner, pitch that idea to them, and if they agree, then we can continue on with the application process. Then it's the 95% success rate. Okay, so the hard part really comes at the beginning. The other thing too that's important is that there are no deadlines, so you can apply whenever you like. So some people think it's just, in September or January that you can start a project. That's not the case. You just need to apply about 12 weeks before you want to start the project. So that's the number that's important. Now, the way that the funding works, it's in something we call units or blocks of funding. And so essentially the maximum amount of blocks of funding you can request are on the screen here. So for undergrads, it's up to 1.5 years of funding. For the master's, it's up to 2 years. PhD, it's 4. Postdoc, it's 3. And if you're a recent grad, you can apply to fund it to get funding from MyTax within the first 2 years of your graduation for up to 2 years of funding. Now, these units of funding or these blocks of funding are basically how you calculate the duration of your project or the total amount of funding you're going to request. So every block of funding is 4 to 6 months in duration, and it has a value that I'll show you on the next slide. And then if you want to build a project that's bigger than 4 to 6 months, you can just stack these on top of each other like a bunch of Lego blocks, and then you can say, I need 1 year of funding, 2 years of funding, et cetera, in that scenario. Now, if you remember at the beginning, I said we want to help you build your CVs. So how do we do that through this program? It's not just the funding, but it's that we expect you to collaborate with the partner. So you need to spend at least 25% of the project working with the partner to fulfill the objectives of the project you've submitted. So this isn't a job. This isn't an internship in the typical sense where you're working in the evenings or you're working on the weekends. This is really integrated into your degrees. Okay, so it's really work integrated learning. You are going to write the project out with your professor, with the partner. You'll lay out the objectives, and then you just split your time between the partner site, between your professor. This can be done virtually as well if needed to fulfill the objectives of the project. So there you're gaining skills that you're going to be able to put on your CV for whatever your next step is going to be. Now, the way that the funding itself works, so every block of funding every 4 to 6 months is split between us and the partner. So say this block of funding is $15,000, so they can be 10, 15, or 20. Let's take 15. We are going to put half, and your partner is going to put half. So when you approach the partner and pitch your project to them, not only do you want them to be an active collaborator, but you want them to put half the money in. So you have to be very convincing. Okay, so you have to show them what the return on investment is going to be by working with you. Now, who's the eligible partner? People ask us all the time, do we have lists? Can we give you a list of partners? We can't just because we don't have memberships. So it's not that people have to be members of MyTax in order for you to be able to work with them. It's anybody who's eligible, so who follows these criteria. So the first one, any company, okay? Any for-profit company in Canada is eligible. Whether it's domestic or foreign owned, we don't care. As long as they're based here, you can work with them. It doesn't matter if there are 2 or 3 people, employees there. It doesn't matter if there are 2,000. Okay, so the size doesn't matter. It doesn't matter how long they've been incorporated. As long as they're a real company, you can work with them. For non-for-profit organizations, a lot of them are eligible, but you need to check with us first. Okay, so there are certain criteria we need to check. So if ever you want to work with a nonprofit in Canada, just send me their name. We will check for you and let you know if they're eligible or not. Canadian hospitals and municipalities are eligible. So this is another one that is usually very interesting for people. They don't realize that these can be eligible partners. So you can collaborate with a hospital, let's say, on a particular project if that project will impact the hospital directly. And then what's not admissible are federal and provincial governments. And the reason for that is that that's who funds us. So we can't double dip funding. Okay, so if it's let's say Health Canada, they're not eligible because they're funded by the federal government. Okay, but again, whenever you're not sure, just please reach out. Happy to help you with that. Now, the other program, very quickly, is the Global Link Research Award. This may be of interest for those of you who would like to go abroad and work with a professor in another country. So this is a travel grant, $6,000, and it's call-based because there are certain allocations that McGill has. So there are 3 calls per year that McGill has for this program. The second or third one, depending how you count, of the year is open right now with a deadline for October 5th. Okay, so the idea here is that you're collaborating with your McGill professor and a professor abroad in one of our partner countries on a 12 to 24-week project. You're basically going to go abroad, do the project, and come back. This one cannot be done virtually. Okay, So if this one is of interest, come let me know. I actually have a kiosk for those of you who haven't come to chat with me, so if you have any questions after, I'm happy to chat with you. But this one is a little bit time sensitive because you do have to apply 16 weeks before your travel date, plus the time it'll take for McGill to pick who will be allowed to apply. So really you're looking at about 24 weeks in advance of applying. Okay, so you really have to plan ahead for this one. Now, just ending with a few resources for you, because I know that these questions will come up. So the first one is, well, I don't have any partners. I don't know how to find them. Can I have that list? So we do have a list on our website of all our previously funded projects that are not confidential. So you can always use this resource just to see who's done what, with whom, get a few ideas on what types of partners exist, and you're not restricted by those partners at all. You can do whatever you like, but it's a source of inspiration. So if you just Google My Tax Projects, you will have access to that. The next one is our open projects page. So sometimes some people will say, I don't have a project to pitch. I don't really care what I work on. I just want to do something different. In that scenario, we have partners who reach out to us all the time looking for specific expertise. We post those online. And so essentially you can go on every day, the list will be updated every day, and see if there are any projects that fit your interests. And if ever you find one, you can just let me know or just hit apply directly on the website. And so in this scenario, you're doing a project that a partner has already decided they need expertise for, rather than pitching your own project to them. And then the last slide is just to show you some examples of projects that we have funded at McGill from the IPN group. These are just 9 projects. There are a lot more. So these are the types of projects that you'll see on our projects page. So if you go on the projects page, you'll get to see who the partner is, what's the title of the project, the lay abstract, the prof, the student. You'll have all the information there. Okay, so you can see the breadth of the types of projects that have been done. And with that, I'll leave you with a contact page. So like I said, I'm Noha. I'm responsible for the faculties of medicine and science at McGill, and I have 2 other colleagues who cover the rest of the faculties. So feel free to take a picture of this, contact any of us as needed. And I have a kiosk out back, so come to see me. I'll be here till about 2 or 3 p.m. today. If you have questions, happy to take them. Thank you. Any questions? Okay, thank you.
Okay, thank you so much, Noha. Yeah, please visit her booth and chat with her a bit. So I guess next up is Crystal Van Hoff, who's the Managing Director and CEO of HBHL. We've all benefited a lot from having HBHL activities. So you don't need sound, right?
No.
Okay.
Everybody. So I am Crystal Van Hoof, and I'm the Managing Director of Healthy Brains, Healthy Lives. And I'm here to just talk about, I'm not going to talk too much about HBHL today because I think you can all learn about that on our website, and I don't have a lot of time, especially now that I've taken up your time with my IT problems. So today I just want to talk about some top sex and gender-based analysis plus errors that we find, well, all over the place, but particularly in the HBHL fellowship applications. That's what I think is most relevant for all of you. And I had to do a lot of reviewing of this this summer because we had someone off on sick leave. So I learned a lot about this. I mean, I know a lot about it at SGBA, but about the errors. So I'll tell you just about our process and why this is important. And the reason is that we first do an external review. Well, we do an administrative review like everyone does, make sure you crossed all your T's and dotted all your I's. Then we do an external review where a minimum of 3 reviewers will review your application. And then we get that back. We decide who's going to get funded and who's going to be our backup applications. And at that point, the fundable applications go out to experts in sex and gender-based analysis to review specifically your sex and gender-based analysis section. And so when that comes back, they will either rate the sex, gender, and other intersectional factors as acceptable or unacceptable or not applicable, because sometimes it isn't applicable. So at that stage, if anything has been rated as not acceptable or insufficient, then we will send it back to you, and the bottom line is that you will not get funding until all 3 of those sections is either acceptable or not applicable. So this is why this is really important for you to know about. So today I'm just going to quickly make sure that we're all on the same page when it comes to terminology, because that's super important, and I still run across some issues there, so that's super easy. And then the second is I'm just going to give you 2 examples of errors that are the most common errors that I see, and what you can do to fix those errors so that I'm not going back and forth with you when you're just waiting to get your fellowship and making you deal with this. So the caveat here is that I'm just for time's sake, going to be doing the 101 version. So there's a lot of things that I could talk about, like that sex is not binary, that sex and gender are difficult to tease apart. You don't always know if you're looking at the biological or the social or combination of both. What a lot of researchers do is if you're not sure, they refer to sex/gender, but that's story for another time. And gender is only one of many intersectional factors affecting health and wellness in our identities. So I'm not going to talk about intersectionality today because, again, no time, because we don't have all day. So terminology. Number one, sex refers to biological differences between males and females like genes, sex, hormones, physiology, and immune response. If you are talking about sex, if you use the word sex, that is what you should be talking about. And gender refers to psychosocial characteristics and factors that affect women, men, boys, girls, and gender diverse people. And so the bottom line here to remember is that every cell has a sex, every person is gendered, and that mice do not have genders. So please, if you are doing animal models, if you are doing cells, please stop telling me about their genders in your applications. Thank you very much. Okay, so unless you've learned to talk mouse, which is fine. Please let me know about that. That would be great. Okay, so 2 common errors. I'll give you one in the biological sex area, and I'll give you one in the gender area and how to address it. So error number 1 that I hear a lot is I will not look at sex because there are no sex differences in XYZ, whatever it is that you're studying, identified in the literature. So why is this an error? Well, if the research has not historically accounted for sex, then the literature is not a reliable source of data on this subject. And spoiler, I can tell you that the research has not historically or adequately accounted for sex. So just about any area that you can look at, it historically has not appropriately accounted for sex. Even if that is increasing with time, it's still not where it needs to be, and we still have a lot more data on males than we do on females, or we don't know who we have the data on because it wasn't reported. So here's just a couple of examples. So this is from Jeff Mogul, who's here at McGill, did 2 reviews where he looked in 2005 and then again in 2006 just at pain research and found all that blue there is the articles that he found that only reported having studied males only, and the orange, they studied females only. The gray, sex was not reported, and usually we can assume that the gray, where it's not reported, is males. And then the little bit of yellow at the top there is where they studied both sexes, and he actually goes into an additional layer of detail to say even when they studied both sexes, a lot of them don't actually even disaggregate their data by sex. So it's, let's say, helpful, but it's step 1, and we need more steps there. So what can you do? So there are, about this particular example, right? So you can study both sexes in your methods, you can include both sexes, you can study them in cellular work if you're using cells where you don't know sort of where they came from, you can find out the sex of their cells, there's actually lists online. You can disaggregate your data by sex, which is super, super important, and look for differences. When you report, it's super important, and there's lots of guidelines out there, and journals are more and more enforcing this, is to report the sexes that you studied. Don't say in mice, say in male and female mice, say in male mice if that's what you're studying, and there are good reasons to do that. And then, of course, report sex disaggregated data when you are writing your article. That's really important, and it helps others who want to look at the literature and see if there are reported sex differences. If you don't report that you found no sex difference, then nobody's going to know that you found no sex difference, and it's not helping anything move ahead. So error number 2, and this is my gender example, is assuming that the only way to measure and study gender is through gender identity. And I would say that this is actually the majority of the SGBA sections this year that got sent back for people to adjust. It was actually this error that we were dealing with. So people would say, for example, we cannot study gender because we are using existing data and gender identity was not collected. So another example would be our cohort is male only, and we do not expect to find an adequate sample of individuals who do not identify as men to conduct a meaningful subgroup analysis. So why is this an error? So the reason this is an error is that gender is a multidimensional concept. It is not just gender identity, i.e. how you identify as a person. So there are gender roles. This is behaviors, norms, how society expects you to act based on how they read your gender or what sex you are, influences all of your everyday actions. It can influence the careers that people pursue, what they believe is possible for them, their role in the family. All of these things can be affected and have very little often to do with how you personally identify. It's about how other people see you and how that affects how you act and the roles you take on. There is, of course, identity, and that's huge. Obviously, it's your inner sense of self, masculine, feminine, in between, neither, whatever it is. But there's also relations, so interpersonal interactions, how you are treated by other people based on how they read you and your gender and your sex are hugely important. And you can imagine all the ways that that could actually affect your health and wellness, as well as institutionalized gender. So how is power distributed in society across how people read your sex or gender? So many of these things don't actually necessarily relate to how you identify and what your gender identity is. So how could you actually look at those things and their effect on health if you didn't have a gender identity measure on your survey that you used in your research, right? So why is this important? I mean, for males, we know that strict adherence to traditional masculine norms can actually be a risk factor for suicide ideation. That's not gender identity. That's just how I think of myself as what it means to be a man, right? And if I hold strictly onto a certain idea of that, then it means that any time that I sort of sway from that strictly held idea, it can create cognitive dissonance, it can create all kinds of wellness issues for people, and that's not gender identity necessarily, right? So that's something that can be measured. Health outcomes can be negatively affected, and we've seen this in many studies, by an individual's level of housework and caregiving responsibilities in the home, which disproportionately fall on women in our society. So that's something that's interesting to look at that is a gendered effect, but isn't necessarily to do with gender identity. So what can you do? So in the example of our cohort is only male, just using that as an example, so consider using an adherence to gender norms inventory in your study. So let's say you have a cohort of male prostate cancer survivors. The study or the intervention is an exercise program. So the question is, does adherence to masculine norms act as a barrier or as a facilitator to program success? And then if you find that it does affect program success, then how might you actually adjust the program to be more effective for a broader audience, whichever group it is that it looks like it's not working as well for? You might be able to adjust your program to account for that. So in the example of we cannot study gender because we are using existing data and gender identity was not collected, what could you do? Well, you could disaggregate your data by sex, because sex is almost always collected, even if gender wasn't. And then you could look for non-biological, look for any differences, but you might find differences that are probably not biological. And so you could call that potentially a gendered effect in your study. So let's say that you have a cohort of males and females in post-op recovery, and the intervention is on the effects of a post-op recovery program, and you could ask the question, did men or women adhere to the program better or recover faster than one another? And then if so, if you did find a difference when you disaggregated by sex, don't just assume that you're finding a biological difference to post-op recovery for this surgery. You need to ask other questions to determine whether you're seeing a sex effect or a gender effect, or maybe a combination of both. So you could ask questions like, did it make a difference what their relationship status was? Did it matter if they were in a heterosexual relationship? Did it matter that they had caregiving responsibilities? Did household or personal income matter? Did neighborhood, and you could use that as postal code, zip code matter? These are all things that may or may not have been collected in the original data that you're using, and you wouldn't have necessarily seen these as gendered, but if you use sex as a dependent variable, then all of a sudden you might be able to create a gender variable to look at how gender might be affecting the intervention and study of their cohort. So something interesting to think about. And you can learn lots more about that because the other thing that you can do is to take all three of the CIHR Institute of Gender and Health training modules. And what I was just describing about gender very quickly there, you learn all about that in course number 3, Sex and Gender in the Analysis of Secondary Data from Human Participants. And I recommend anyone that's dealing with human data to take that course, because it's really good about showing you just sort of problem solving through different ways that you can create gendered variables when you don't have specifically collected gender variables. And that's it for me. Thanks very much. Thanks.
That's good.
I'm not sure. Okay, great. I'll grab that. Thank you very much, Crystal. So next is, you don't have
I don't have a
Okay, so Roberta's going to be talking about EDI committees at IPN and
Yeah.
kind of a...
Yeah, I don't have a
Annual
presentation because I really thought about talking to you like that. So, hi everyone. I'm presenting the Equity, Diversity and Inclusion Committee, which was established last year at this time of the year, more or less. So the committee is actually, it actually includes 3 different subcommittees. One, because we wanted really to cover all the aspects of EDI. So the first committee is the Systems Committee, which is chaired by Austin Milnerwood, and the secretary is Erin Wall, in case you know them. And with this committee, we would like really to tackle how IPN works from the recruitment process to the progress of students to the graduation. There's a second committee, which is the one that I'm chairing, which is called the Knowledge Committee. And with this, our aim is really to increase the knowledge of EDI principles and application of them, and spread awareness on EDI policies to all the people involved within the IPN program. The secretary is Lou Messier. The third committee is the Empowerment Committee, and it's really project-oriented. They really work to empower people from historically marginalized and underrepresented groups for all the life throughout the IPN, their IPN program. So even starting with the application process through the recruitment and how they live during their graduate studies. So in order to do all these, of course, these 3 subcommittees are very much interconnected. We try as much as possible to work one with the other because, of course, one depends on the other way of working. What we are doing right now is that we are at a phase in which we are collecting data based on surveys that have already been collected, but also we will create our own surveys. So in the next months, hopefully you will receive surveys to fill in. So that's what I wanted to tell you today. For more information, you can reach out to me or the other chairs and secretaries. I forgot to say the System Committee chair is John Sakata, and the secretary is Marina Tedeschi. You can look for me. I'm around today till 2 p.m., or you can look for me online. I believe I'm the only faculty named Roberta in the IPN program, so that's pretty easy. Thank you.
Thank you, Roberta. So the activities of these committees they're sort of rapidly ramping up, and we're going to be trying to work on organizational collaboration between the different committees over the next few months. So I think if people are interested in getting involved in the EDI committees, I strongly recommend now is a good time to contact them. Okay, so the next session is a symposium session on early life adversity. It's going to be run by Naguib Meshwar, and I'll put up the slides. Here we go. Okay, thank you very much, Naguib.
So hello everyone. As Ed said, I'm Naguib Meshwar. I'll be chairing this morning's symposium, but first I'd like to acknowledge and thank Ed, the IPN team, and all the student volunteers for their wonderful work in organizing this very successful retreat. So thank you. So yes, this symposium will be focused on early life adversity in humans, and I thought that I should maybe give some definition, just one slide about early life adversity itself. What does it mean? And the talks you'll hear during this symposium will be focused mainly on childhood abuse, a very common form of adversity. And I have a few bullet points here. What do we mean by childhood abuse? Well, it's abuse, it's maltreatment perpetrated typically by parents or caregivers who are very close to the children. And it affects children of all ages, economic and cultural backgrounds. It's very widespread. I have prevalent statistics here that are quite conservative. They concern mainly severe and chronic abuse. And I think Claudia will mention some statistics that are higher than that from a Canadian study. And what's quite dramatic about childhood abuse and maltreatment is that it strongly predicts very poor outcomes, so both socioeconomic and health outcomes. So there's a plethora of studies showing that abuse is early in life is associated with educational underachievement, with increased crime, unemployment, and also there are adverse consequences in terms of health. So obesity, cardiovascular disease, chronic pain, these are just 3 examples out of many. And obviously, we are interested, and the students who will present are interested in the adverse consequences with regards to psychopathologies. So again, many studies have associated a history of childhood abuse with mental illnesses like post-traumatic stress disorder, and it's very common to see also very high rates of depression, major depressive disorder. Suicide, unfortunately, is a far too common consequence of a history of child abuse. Now, just to show you how impactful childhood abuse can be and how cumulative it can be, because there are different forms of abuse, of maltreatment. I haven't told you yet what they are, but you've guessed it. So psychological abuse, physical abuse, sexual abuse, neglect, and many other maybe a bit more minor forms, but there are different types of maltreatment. And again, many studies have shown that there can be a cumulative a negative impact of maltreatment. So when there are multiple forms of maltreatment during childhood, it has more adverse consequences, so higher rates of mental illness, of suicide, for example. And so the graphs on the right-hand side are from Martin Teicher's group at McLean's Hospital, Harvard, recent paper, in 2015. This is using self-assessment questionnaires in young adults. It's a study of about more than 500 young adults. And you can see, I think, quite clearly on the top you have scales for depression, on the bottom for suicidal ideation, and there's some sort of linear relationship. The more forms and types of maltreatment, the more severe the symptoms are. So it's a big problem in all societies, and what the symposium is about is to try to identify some mediators of this increased vulnerability from early life adversity. So without further ado, we have 3 outstanding speakers, all PhD, IPN PhD students based at the Douglas. So Daniel Almeida, who's finishing his PhD in the Tureki lab, Claudia Belliveau, who's in my lab, Ashita Batra in Patricia Silvera's lab, and finally Alice Morgunova in Cecilia Flores' lab. So, Daniel. Thank you.
Just got to put it up a bit higher. So as Naguib said, my name is Daniel, and I'll be presenting some of the work that we've been doing in the Tureki lab on childhood abuse-associated epigenomic and transcriptomic differences in the prefrontal cortex. So Childhood abuse is a highly prevalent global health concern that increases lifetime risk of negative mental health outcomes, including depression and suicide. In both children and adults, a history of childhood abuse has been consistently associated with structural and functional alterations to the PFC, as well as its connectivity to downstream structures. One of the questions is, how is it that early life adversity leads to these long-term consequences? What we know is that childhood is a sensitive period during which the brain is more plastic and responsive to the environment. Epigenetic mechanisms may actually contribute to this plasticity, and so we think that epigenetic mechanisms may serve as that molecular link between early life adversity and long-term consequences. To date, however, only a handful of postmortem investigations have been conducted in order to characterize genome-wide changes to the epigenome in the context of child abuse. However, of these studies, most have actually been conducted with tissue homogenates. One of the issues with tissue homogenates is that they mask cell type specific changes associated with the history of childhood abuse. So what we wanted to do is really try to understand on a cell type specific level, what are the long-term changes that occur to the epigenome in the context of childhood abuse. When thinking about the cell type of interest, we chose to focus on deep layer pyramidal neurons, and that's because of their importance to PFC microcircuitry and their connectivity to downstream structures involved in the regulation of mood and emotion. So we had two major aims for this project. The first was to first develop an isolation method that would allow us to isolate deep layer pyramidal neurons from postmortem human brain samples, and then finally to identify those genome-wide patterns of both differential methylation and expression in these neurons in the context of childhood abuse. So before I jump into those aims, I'll talk a little bit about our cohort. So we had subjects who had a history of severe childhood abuse, who then developed depression and died by suicide later on in adulthood, and then control subjects without a history of childhood abuse, who were not depressed and died by accidental causes. So I'll start off with our first aim, which is the development of that methodology for the isolation of deep layer pyramidal neurons. So what we did was we turned to a fluorescent activated nuclei sorting approach to allow us to separate out these cells. We used a combination of nu-N for the isolation of neurons, and then CTIP2, which is a transcription factor that's involved in the development of deep layer neurons. Now, what you'll notice on the graph on the right-hand side is that 3 populations were identified in order to go deeper into our cell type of interest. We also gated based off of forward scatter. So forward scatter is proportional to nuclei size. What we know is that deep layer pyramidal neurons have the largest nuclei in the PFC, and so by using forward scatter, we can actually select specifically for the pyramidal neurons that we were interested in. Now, once we actually had our cells, you can imagine that isolating a specific cell type means that we don't have a huge concentration of genomic DNA that you're able to isolate. This is a comparison of previous postmortem investigations that have done whole genome bisulfite sequencing. And what you'll notice is that the inputs of genomic DNA are quite large. This was a major challenge for cell type specific sequencing. And so what we did was we turned to a newer methodology in the field of epigenetics, which is enzymatic methyl sequencing. This technology is a lot softer on your genomic DNA. Bisulfite tends to be quite harsh and ends up leading to some artifacts in sequencing. And so by turning to this methodology, we're actually able to use an input of 20 nanograms. And this substantially opens up the possibility of doing more cell type specific epigenomic profiling in postmortem brain samples. So just looking at the quality of our data, what was I think most surprising was that our duplication rate, which is usually an indicator of the quality of genomic DNA that you're inputting, was actually quite low and in fact superior to what we would see with a bisulfite-based approach. We also showed that the coverage across different trinucleotide contexts was equal, suggesting that this approach didn't bias against any particular genomic context. We then wanted to confirm that our sort was pure, and we found a enriched population of course, deep layer pyramidal neurons using bioinformatic deconvolution and a single nuclei methylation sequencing data set. Looking at genome-wide levels of DNA methylation, of course, as expected, the highest levels were in the CG context. And then what you'll notice is that all 12 other contexts would be representative of non-CG methylation. And as expected, the highest levels were in the CAC context. This has been reported in both mouse and human. So I'll just talk a little bit about non-CG methylation before I delve into our data. Non-CG methylation is particularly abundant in the neural epigenome. In vertebrates, non-CG methylation is enriched in highly conserved developmental genes. And unlike CG methylation, non-CG methylation is negligible in the fetal brain and actually develops quite rapidly during the first 5 years of life. And this parallels primary phases of synaptogenesis. Non-CG methylation occurs most commonly in the CAC context, and so it's unsurprising that we see these high levels. So we then wanted to look at whether or not there were sort of genome-wide changes to methylation across trinucleotide contexts in the context of child abuse, and we found that this was not the case. So we then moved on to investigating differential methylation and expression in our deep layer pyramidal neurons associated with the history of childhood abuse. So what I'll turn your attention to here is using DMR-seq, which is a program that allows you to identify regions of differential methylation in case control studies. What you'll notice is that the number of CAC DMRs that we got was substantially higher than the CG context. In order to make sure that this wasn't just because of non-CG methylation, but rather CAC specific, we also looked at CTC context as well. And what you'll notice is that the CAC context still stands out as really leading to quite a few differentially methylated regions. This might suggest that CAC methylation is particularly sensitive to the effects of child abuse, and that makes sense because CAC methylation is accumulating at the time that these individuals are exposed to the adversity. So we then assigned each of our DMRs to different genes and asked whether there were particular biological processes that were enriched. And what we found was synaptic terms that came across the board across all DMR types. Okay, well, the slide's missing something, but what it was supposed to show was that CAC DMRs actually regulated largely unique genes. So it's not the case that these sort of greater abundance of DMRs are located in the same genes, but rather are regulating a diverse set of genes that are not regulated by the CG or CTC DMRs, again pointing to CAC methylation being particularly sensitive to the effects of child abuse. We further wanted to characterize more on a genomic level some of the differences between each of our DMR contexts. And what we found is non-CG methylation contexts, that is to say CAC and CTC, were quite different from that of CG methylation. Also looking at the distribution across chromosomes, this was also the case where non-CG methylation was quite different from CG methylation. And then finally, looking across different genomic features, this was also the case where CAC and CTC methylation were quite different from CG methylation. So altogether, these data suggest that non-CG methylation may respond differently to a history of childhood abuse with respect to the distribution across chromosomes, distribution across genomic features, but that all three types are responding to a history of childhood abuse by impinging upon the same biological processes. So the next thing that we wanted to do was to ask whether or not these DMRs actually have a functional impact on gene expression. And so to do that, we had to isolate deep layer pyramidal neurons. We did so using laser capture microdissection based off of their pyramidal morphology and large apical dendrite. You can see in the image on the top. Before actually looking at gene expression, we wanted to integrate the two data sets to ensure that they agreed with one another. And what we showed was that methylation showed that typical anti-correlation with gene expression. More methylation, less gene expression. So looking at differential expression independent of our methylation data, what we found was differential expression of 281 genes. And again, we saw an enrichment of synaptic-related processes. In fact, 54 out of our 281 genes were considered synaptic-related, and altogether this is suggesting that there is strong agreement between our methylation data and our expression data in deep layer pyramidal neurons. We then looked at the overlap between our differentially methylated regions and our differentially expressed regions, and quite a few of those differentially expressed genes harbored differentially methylated regions. This was particularly the case, again, for CAC methylation, where we saw more of our differentially expressed genes regulated by CAC DMRs. So I'll go ahead and summarize our two aims. The first is that EMSeq produce high-quality data that's comparable, if not superior, to the gold standard in epigenetics, which is bisulfite sequencing. For AIM2, despite the stark contrasts that we observed for CG and non-CG contexts, the two contexts seem to respond to childhood abuse by affecting genes involved in synaptic processes. However, non-CG methylation, specifically CAC methylation, seems to be particularly sensitive to the effects of childhood abuse, and this may be because of the temporal dynamics, that is, the accumulation of CAC methylation during the experience of the adversity, whereas CG methylation tends to accumulate during fetal development. So importantly, these epigenetic changes were mirrored in the transcriptome, suggesting that they have a functional impact on gene expression and a functional impact on the PFC. So I'd like to thank my supervisor, Gustavo Trecki, Naguib Mechwar, and Cecilia Flores for their insight throughout the project, and then, of course, the McGill Group for Suicide Studies, many of which are here today, the Douglas Bell Canada Brain Bank, where our brain samples are from, the Douglas Molecular and Cellular Microscopy Platform, Chino, Quebec, and our collaborator, Dr. Matthew Sutterman at the University of Bristol. I'll take any questions.
Hi, Daniel. Thank you for the great talk. So my question is, when you collect data from people who died by suicide with childhood abuse, in these cases, are you able to tease apart how many of them had access to therapy or like other things, like if it was an isolated incident or prolonged abuse, or is it just, okay, abused or not abused?
Yeah, so the way that we categorize abuse is through the SICA, so the childhood experiences of care and abuse. And we're looking at specific cases of severe abuse. So whatever that definition is for the SICA, this severe cases of childhood abuse. I don't think that we collect data on whether or not participants were in therapy. We do have different types of abuse, but that can be challenging for power issues, for trying to understand and teasing apart sort of subtypes of abuse and the effects that it has on the brain.
Is there anything known about reversing these effects over time with like intervention, or is it once this is done, you can't reverse these changes?
It's definitely an interesting question. I think some of the literature points to what's important is sort of the cumulative effect of trauma throughout life, as opposed to just one sort of experience of childhood trauma. Certainly, animal models have been used where an early life adversity paradigm is used and then environmental enrichment later on. And that does seem to influence some of the findings, but I'm not sure that it's well characterized in humans.
Okay, thanks.
Thanks.
Hi, Daniel. Great talk. I was wondering if you guys looked into maybe reversing some of the epigenetic changes, or if you guys have any sort of next steps that I'm just curious to know.
Yeah, so one of the great things about postmortem human brain samples is that we're really studying childhood abuse in human subjects, but postmortem brain samples are really a snapshot of a really complex picture, so it's difficult to tease apart really the temporal dynamics of the effects and whether or not they're reversible, and I think animal models are probably important in this context.
Hi, thank you for your talk. I was just wondering, do you have like a cohort of genes, like to what pathways do they belong to, the genes that are modified with these epigenetics modifications? I just wanted to know how their function might impact later on what's going on in the brain of these individuals.
Yeah, so a lot of the genes that came up, there was enrichment for synaptic-related processes. I think this is particularly interesting because a lot of the brain imaging literature shows that the connectivity between the PFC and other brain areas involved in the regulation of emotion is consistently impaired across the board for subjects with a history of childhood abuse. So I'm sure that that might be sort of one implication of these changes to synaptic-related processes.
Hi, my name is Claudia, and today I'll be presenting the lasting impact of child abuse on perineuronal nets in the ventromedial prefrontal cortex. So child abuse is defined as physical, sexual, or emotional maltreatment or neglect of a youth. And unfortunately, 1 in 3 Canadians has experienced some form of maltreatment before the age of 15. Now, we know there's a lot of literature that points to functional and connectivity differences in individuals that have experienced child abuse. And generally, these differences are, I'm just going to turn on my laser pointer. Generally, these differences are in parts of the brain that are connected and function in threat regulation. Interestingly, with 300 million people experiencing major depressive disorder globally, those with a history of child abuse are more likely to have more severe symptoms, they're less responsive to treatment, and they have more recurrent episodes. But the question is why? What is happening in the brain during the abuse that leads to these negative consequences later in life? We also see an increase in outcomes of suicide in individuals with an experience of child abuse. So we know that childhood and adolescence are marked by a critical period of plasticity. So this is a time where the brain is easily influenced by the outside world, with synaptic connections being able to be modified and rearranged depending on the environmental input. This opening of these critical periods occurs because of the maturation of fast-spiking inhibitory interneurons. The closing of these periods happens because of multiple occurrences, but what's important to us is the development of these things called perineuronal nets. So these are structures that sort of harden circuitry and stabilize connections between different cells in the brain, leading the brain to be less plastic. So perineuronal nets are a condensed form of extracellular matrix that form around the cell soma and proximal dendrites of certain neurons in the brain, mainly parvalbumin-positive inhibitory interneurons. And so these structures are a mesh-like network of chondroitin sulfate proteoglycans like aggrecan, versican, neurocan, and brevican, which form around a hyaluronan backbone and are connected to each other with link proteins and tenascin R. So these nets develop from early childhood through late childhood, and they seem to sort of stabilize in number into adulthood. So they really are a structure that concretizes connections in the brain and last into adult life. So perineural nets have multiple functions. They serve as a physical barrier between a neuron and the extracellular space, as well as they allow for binding of specific proteins. So interestingly, OTX2, which is a factor that's very important in parvalbumin-positive inhibitory interneuron maturation, actually binds to PNNs first before it goes into the cells. They also are known to limit mobility on the neuronal membrane, so keeping AMPA receptors in place, for example. And they're known to stabilize some forms of memory. So specifically, perineural nets are thought to protect fear memories from erasure. So animals, adult animals that have experienced fear conditioning usually have fear memories that are resistant to extinction. And if you actually chemically reduce the number of perineural nets in the amygdala in an adult animal, you can then reduce and extinct the fear memory. So it really, if you delete these nets, it reopens this plasticity of the brain. So our general hypothesis is that child abuse occurring during this critical period disrupts cortical connectivity through altered recruitment of these perineural nets in brain areas involved in the control of emotion in depressed suicides. So we look specifically at the ventromedial prefrontal cortex. So I'm very privileged to work with the Douglas Bell Canada Brain Bank, where we have access to post-mortem human brain samples. And I look at 3 different groups. The first being the group of interest, which is the depressed suicides with a history of child abuse. So as Daniel and Naguib mentioned, these are individuals who have been diagnosed with depression throughout their life and have chosen to die by suicide. And then through psychological autopsy, we see that they experience severe abuse. We then have sort of 2 control groups, one being psychiatrically healthy controls who have no history of neurodevelopmental disorders, neurodegenerative disorders, or other mood disorders that have died accidentally. And then a third group of depressed suicides, so people who were diagnosed with depression but have no history of child abuse. And so we have a cohort of 39 samples. And we sectioned their brain tissue. We performed immunohistochemistry or fluorescent in situ hybridization, imaged the slides, and then analyzed them using QPath or Fiji. And so very interestingly, we see that after looking at the densities of perineuronal nets in the ventromedial prefrontal cortex, we see that there is a significant increase in the density of PNNs in layers 3, 4, 5, and 6 of the VMPFC, specifically in the depressed suicides with a history of child abuse. The next thing we looked at is the intensity of the staining of these perineuronal nets. So in the literature, this is used as a proxy to sort of understand how long the PNNs have been around, so how early they developed or how active the cell beneath it really is. And so you can see here that in the same sample, you have a PNN with low staining intensity and a PNN with high staining intensity. So we took the average intensity and we found that depressed suicides with a history of child abuse actually have much brighter PNNs, as well as, on average, the PNNs cover more of the cell. So they're more complex. They go more onto the dendrites. So all of this is sort of suggesting a precocious maturation in these individuals with child abuse. So the next thing we wanted to do is we wanted to see whether these extra nets are forming around all cell types or specific cell types. So the first thing we did was characterize for the first time in postmortem human brain the identity of cells that are covered by perineural nets. So in controls, we found that around 75% of perineural nets are covering parvalbumin-positive inhibitory interneurons, so much like the rodent literature. And in fact, we found 23% of these PNNs are covering other inhibitory cells, so GAD1 positive inhibitory cells that are not parvalbumin positive. And lastly, what's really interesting is we found that 3% of perineural nets are actually covering SLC17A7 positive excitatory cells. So this is really interesting. So we looked then by group, and we looked at the total number of excitatory, inhibitory, generally, or parvalbumin positive inhibitory interneurons. And we actually see that PNNs are increased solely around parvalbumin positive inhibitory interneurons. So this sort of increase in nets is really around this cell type. And then we were wondering, okay, is it because there's more PV cells for more nets to form around? And in fact, we don't see a difference between groups in the density of the PV cells. So really, there's an increased recruitment of these perineural nets. So then the next question we wanted to answer was sort of what's happening here? How is this happening? So we used a previously published data set from our collaborators in the McGill Group for Suicide Studies that did a single nuclease RNA sequencing, but in BA9, so still prefrontal cortex, but not the VMPFC. And we looked at the expression of certain canonical PNN components in different cell types. So in looking at these canonical PNN components, we see strikingly that a lot of these components are highly expressed in oligodendrocyte precursor cells. And so previous research from our lab has already showed that child abuse does actually affect myelination and OPCs in the postmortem human brain. So this was very interesting. So we went back to our VMPFC samples and we performed fluorescent in situ hybridization for PDGFR alpha, which shows you OPCs, and then some canonical markers that were highly expressed in VCAN, like VCAN and PTPRZ1, which is a phosphocan gene. And so we see that, in fact, even in our VMPFC samples, that a very high percentage, so around 97% of VCAN positive cells were OPCs, and about 91% of PTPRZ positive cells were OPCs as well. And so we also found during this experiment that we saw some OPCs that were directly juxtaposed to these parvalbumin cells. So that was also very interesting to us. we then looked at the protein expression of Versican, and we saw that although the mRNA is expressed in OPCs, we do see that the protein expression is indeed overlapping with WFL-positive perineuronal nets. So hopefully showing that OPCs are producing these canonical PNN components that are then being translated into proteins and packaged into these perineuronal nets. So then we wanted to look between groups and see if there was any differences in these canonical PNN components. And we saw that Versican, Phosphocan, and TNR are all significantly upregulated in OPCs in child abuse group. So the last thing we sort of wanted to understand more of is whether there was a link in this Versican TNR expression and in PNN density. So we, in fact, see PTPRZ1 and TNR expression levels are highly correlated with PNN densities. We also see that on average, OPCs are closer to parvalbumin-positive inhibitory interneurons in the child abuse group. So all of this really comes together and packages and shows us that OPCs may be implicated in this upregulation of PNNs in the ventromedial prefrontal cortex of depressed suicides with a history of child abuse. So just some leading questions that we have ongoing in the lab are what is regulating this increase in nets and the maintenance of these PNNs? So literature has actually recently been implicating microglia, the resident immune cell of the brain, in regulation of these nets. So whether it's direct pruning of the net or the secretion of proteases that can then digest the PNNs. And so I'll just show a little bit of ongoing work. So what we've optimized is staining for perineuronal nets and microglia, and we're going to be looking at the sort of proximal association of the two cell types, so are they closer or farther, the densities of these cells. We also are interested in looking at the activation status of these microglia, so whether there are group differences between expression of CD68, which is an activation marker for microglia, between groups. And lastly, we've already found that there is a significant decrease in MMP9, so a canonical MMP that's very oftentimes implicated in PNN degradation in the literature. And we see that there is a decrease in depressed suicides with a history of child abuse. So thank you to everyone in the MGSS. Thank you to all my coworkers, and thanks for having me here today at the IPN Retreat.
Hi, thank you for the cool talk. So my question is, you were talking about perineal nets in connection with the fear response. So is there anything known about, does it's child abuse or something to do with maybe if the person at a young age experienced a natural disaster or something that is an intense incident that might trigger fear response? Would you expect to see perineal nets in any kind of event that involves fear response, or is specific, there's a social component that's more prolonged, like child abuse?
So PNNs are all around the CNS, so regardless of if you have child abuse or not, they're there in control subjects. So whether we would see this increase because of a natural disaster and one sort of incidence, we don't know. We can't really study that in our postmortem human samples. There are animal studies that go through early life stress, like maternal separation, limited bedding, limited nesting, and we do see effects on PNNs as well. So I can't exactly speak to a natural disaster specifically, but I'm sure one day we'll be able to study this.
Okay. Because in your controls, if you, let's say you see now that, comparing the two groups, you identify that in people with childhood abuse, the PNNs look a certain way, or they're present in certain places, But in controls, if you saw a similar profile, like if you see an outlier that looks very much similar to your test group, would you, because sometimes it's hard to get that information for some people, would you exclude those data points that look like your test group, or would you still include it as an outlier?
So generally, we are quite strict with selecting the individuals that are in the cohort. So we have very rigorous postmortem analyses that are done by our psychologists at the brain bank. And so basically, we have very severe cases of child abuse compared to absolutely no history on medical records, social services records, next of kin says there's no abuse, stuff like that. So we really are quite sure that we're having very well characterized separate groups. Although working with postmortem human brain, it's quite heterogeneous between samples, but we did not have any outliers in these sets of data, but I mean, we run outlier tests and the Grubbs test, so statistically we do take into account outliers, but we had none in this situation. Okay. So yeah.
Hi, thank you so much for your talk. Very impressive and also very inspiring to me because I also work on perineural nets. So my question here is that do you have any working models for how perineural nets influence the function of perineural parvalbumin neurons in VMPFC? Since I'm not so familiar with that brain area. Mm-hmm. Do you have any hypothesis or working models how this work?
Yeah, so our sort of working model is that these extra PNNs in this area that's involved in emotional regulation might just be sort of stabilizing maladaptive circuitry that comes to be during the abuse. So any changes in signaling from pyramidal to inhibitory cells that may occur in terms of a coping mechanism during the abuse might be stabilized by this extra recruitment of perineural nets and sort of leads to these long-term negative or different functioning of the VMPFC. Specifically, cellular work on PV cells, no, we can't really do that in postmortem human brain. But there are studies where they, delete PNNs off of inhibitory cells and see their, depending on where in the brain, either their sort of activity increases or decreases. So it is very region specific, but I have not seen any research in the VMPFC, MPFC of animals that really looks at PV firing yet. It's in other nuclei. But
okay, so so the VMPFC is basically to control the emotion or something?
Yeah, it's involved in the circuit regulation of threat response, decision making, so hot decision making, like Dr. Ragsdale talks about. I'm not sure if he's still here, but sort of more impulsive decisions, more decisions that are involved in emotion, and yeah.
Cool. Thank you.
No problem.
Okay. Hi, everyone. So my name is Ashta Batra, and I am a student under Dr. Patricia Silvera at the Douglas. And today I will be walking you through my thesis project where I'm studying how variations in insulin function modulate the effects of early life adversity on executive functioning. So let me start off with some background just to set up the stage. So insulin is known to inform the cell to take up glucose from the bloodstream. And although that is its primary role in the body, insulin is actually not really needed to do that into most brain cells. Instead, there are transporters like GLUT1 and GLUT3 that transport glucose into the brain, which basically leaves insulin to play several different roles, including executive functions like cognition, memory, emotions, reward, just to name a few. And insulin does this by acting on the prefrontal cortex, the striatum, and the VTA, which are coincidentally the same areas and executive functions in which dopamine has shown to play a role. And I was going to go on about dopamine for a while, but I know we're running a little late and my talk is a little longer than other people, so I will skip this part. So the last part of the project that I will be discussing is early life adversity. And so early life adversity can occur prenatally, such as gestational diabetes, substance use, maternal anxiety, depression, and it can also occur postnatally during the early years of one's life, such as low SES, parenting quality, maternal depression. And this early life adversity has actually been shown to affect pancreas development, which leads to insulin sensitivity early in life, leading to brain insulin resistance later on in life. And also, early life adversity has been shown to linked to impaired dopamine-related behaviors, such as executive functions later on in life. So keeping all of these pieces in mind, we hypothesized that the genetic profile that's associated with higher fasting insulin would interact with postnatal adversity, influencing the development of executive functions, such as impulsivity. So we tested this hypothesis, and we published our findings in Frontiers in Neuroscience last year, and I'm going to quickly go through a summary of these findings. Here we're starting off with the genetic profile for fasting insulin by looking at a GWAS, which was conducted in non-diabetic adults. So to account for the polygenic nature of the fasting insulin trait, we created a polygenic risk score, or PRS, where the higher the score is, the more that individual is at risk for having high fasting insulin. Instead of using an arbitrary p-value threshold to calculate this PRS, though, what we did was we calculated the PRS at 100 different thresholds, which I'm representing here with a gradient of colors in this GWAS. And then we correlated each one of those PRSs, which we built from this information from this GWAS performed in adults, and we correlated with peripheral insulin levels in the children in the ALSPAC cohort so that we could find which threshold best predicts peripheral insulin levels accurately within children. And through this process, we were able to identify the strongest and most significant association to be at a PRS whose threshold was 0.24. Then in step 2, we further refined this PRS. We ran a linear regression analysis for each SNP within the 0.24 threshold PRS to find which SNPs were significantly predicting the peripheral insulin levels. So basically, in step 1, we found the combination of SNPs that together were most associated with peripheral insulin levels in children. And then we wanted to know, out of those SNPs, which ones are statistically significant in predicting this association. And we're visually representing this here by a filter. And at the end of this filtering, we ended up with 57 SNPs out of the 10,000 some that we started at the beginning. And then lastly, in step 3, we can now build an RPRS, or refined PRS, in our testing cohort using the 57 SNPs that we had identified at the end of step 2. And before I move on, I do want to take a moment to highlight why this refinement was so necessary that we did. And the number one reason, there's several, but I just want to highlight number one reason is that the GWAS we were using is made in adults, and we're trying to test this hypothesis within children. And genetic markers that are relevant in adults might not be the same ones that are relevant in children for a specific trait. So this refinement process allows us to identify which SNPs are the ones that are actually affecting children in this case. So then to investigate the interaction effect between the RPRS and early life adversity, we calculated a postnatal adversity score for each individual in the cohort. And then we used a snack delay task to test for our outcome of impulsivity, which was performed at 36 months in the children of the MAVEN cohort. For you here who are not familiar with the snack delay task, it's very familiar to the marshmallow task, which I know more people are familiar with. And then to analyze the data, we ran a linear regression analysis and with the intention of testing our hypothesis, which was whether the RPRS for fasting insulin interacts with postnatal adversity to predict impulsivity. So here are our results. The x-axis here represents postnatal adversity, where adversity experience increases towards the right. The y-axis represents impulsivity, where impulsivity exhibited increases as you go down the graph. And the solid and dotted lines represent individuals with high and low PRSs. And so what we see here is an interaction where higher PRS is linked to more impulsivity in children that were exposed to postnatal adversity. And this tells us that the genetic background associated with fasting insulin levels interacts with postnatal adversity to predict childhood executive functions, specifically in this case, impulsivity. So next, we decided to inspect this fasting insulin genetic score in a sex-specific manner because by the time we were done with the study, a new GWAS came out and it was sex-specific, which is great. But we wanted to look at executive function outcomes in this case from more of a broader perspective. So based on our results from our previous studies, we hypothesized that the genetic background associated with this altered fasting insulin would share some kind of polygenic architecture with the genetic background associated with executive functions. So our goal really here was to find SNPs that overlap between the sex-specific fasting insulin GWAS and 2 executive function GWASs that we decided to start off with. 1, an impulsivity GWAS that we built using the UK Biobank, and 2, the ADHD GWAS that is also publicly available online. And to inspect the genetic architecture, we use a technique called conjunctional FDR, which basically identifies genetic markers that are shared between 2 separate genetic data sets. Unfortunately, though, after running this analysis, we didn't find any SNPs in common between the fasting insulin GWAS and the impulsivity or ADHD GWAS. But this lack of shared genetic architecture informs us that although altered fasting insulin is associated with altered executive functioning, the 2 don't share any genetic markers with each other, which solidifies our hypothesis that there must be something interacting with fasting insulin, such as early life adversity, that results in these altered executive function outcomes. So to further inspect this, we conducted a similar study to the first one I showed you to inspect the interaction effect on executive functioning, but this time in a sex-specific manner. So we used the sex-specific fasting insulin GWASs to create our polygenic risk scores. Again, the higher the score, the more that individual is at risk for having high fasting insulin levels. And then, exactly like in our first study, we calculated the RPRS using the sex-specific GWASs separately in males and females. In males, we had identified a threshold of 0.05 and ended up with 635 SNPs. In females, our threshold was 0.15, and the final SNP count was 1,449. And then instead of running a regression analysis for one specific executive function outcome this time, we decided to run an executive function PheWAS that would be predicted by the interaction between the sex-specific RPRS and postnatal adversity. And then we also decided, why not, to add in 4 different cohorts so we could inspect this at 4 different ages. So I'm going to now walk you through the PheWAS results from our analysis. So these are results from the MAVEN cohort. On the left is the male's executive function PheWAS, and on the right is the female's executive function PheWAS. Each dot on the graph represents an executive function phenotype, and the y-axis is the negative log of FDR p-value of the association between the executive function phenotype and the interaction. And remember that the interaction is between the risk for high fasting insulin and postnatal adversity. So the dotted line on the graph represents the significance line with the alpha of 0.05. So at the end of it, the higher the dot is on the graph, the higher the interaction effect on predicting a specific executive function phenotype. And at first glance, I'm sure you can see here that none of these phenotypes crossed the FDR significance. None of them were above this dotted line. But if you further look into these results, you can see that there are certain phenotypes that were specifically of interest to us that are significant before the FDR correction. In MAVEN here, in these results, the phenotypes that stood out to us were attention problems, ADHD, delay in SST, or detection error in SST. And if you look at, went too far, the Gusto results, which are set up the exact same way, the phenotypes of interest that are jumping out to me are attention problems, reaction time, externalizing problems. Here is ABCD, which none of the outcomes really jumped out in males, but in females we have attention and ADHD jumping out again. And then in UK Biobank, which is made up of adults, here we see in males that ongoing addiction to alcohol was significant, and in females, depression was significant. And this is interesting because attention and impulsivity-related problems in childhood map to these kind of phenotypes in adulthood. So we see a general trend occurring between all of these cohorts. And this tells us that, again, the genetic background associated with fasting insulin levels interacts with postnatal adversity to predict bunch of executive functions that are related to impulsivity and attention in a sex-specific manner. So next, we wanted to know whether all these associations that we've been seeing so far, do they have any causal relationship with each other, or are they just associations? So we started a collaboration with the Max Planck Institute between our lab and Dr. Elizabeth Binder's lab to investigate the causal association between the sex-specific fasting insulin GWAS and the executive function GWASs that we identified through the PheWAS results that I just showed you. And you do this by using a technique called Mendelian randomization. What Mendelian randomization does, it uses genetic variants, in our case we're using SNPs, to evaluate a causal relationship between an exposure, which for us would be fasting insulin, and an outcome, which we decided based on all our previous results and PheWAS to start with impulsivity as our outcome. Now, before I jump into how we ran our analysis for Mendelian randomization, I just want to review some assumptions that are essential for this method. There are 3 assumptions of MR that you have to ensure are not violated so that you can run this method. First, you have to be sure that the genetic variants that you are using are valid for your exposure. And in this case, in our case, the SNPs that we're using do in fact align and are associated with peripheral insulin levels like I showed you in Al's pathway at the beginning of this presentation. Second, we need to show that the SNPs that we're using from the fasting insulin GWAS do not affect another phenotype that would then be affecting our outcome, also known as confounding variables. So we performed enrichment analyses here that show that insulin and executive functions are in fact associated with each other. With that, in addition to literature review, we can assume here that there are no confounding variables that are playing a role. And then third, we need to show that there is no horizontal pleiotropy, meaning that the genetic variants that we're using do not influence the exposure. The genetic variants that influence the exposure do not influence the outcome through a different channel or method than the exposure itself. So what we want is vertical pleiotropy, where the SNPs we are using affect fasting insulin, which then affects impulsivity. So that would establish a causal association. We have to ensure that something like horizontal pleiotropy is not happening, where the SNPs that we're using are not directly affecting impulsivity and as well as directly affecting insulin. And to me, this seems to be the most critical assumption. And thankfully, there is actually a statistical test you can perform to significantly deny the presence of horizontal pleiotropy. It's called the Egger test. This is important because it will show up in a second. So you have to make sure that you run this Egger test every time you perform MR. So now back to our model that we're using. Since we're interested in investigating the interaction effect, we wanted to run Mendelian randomization in 2 separate groups. One that is exposed to adversity and one that is not exposed to adversity. And we hypothesize here that only individuals that are exposed to adversity will show an association between fasting insulin and executive functioning. So here's a visual representation of the interaction model. Basically, we want to run MR between fasting insulin, our exposure, and a GWAS where individuals exhibit impulsivity and experience exposure to adversity, separately in males and females. And then separately, we want to run MR between fasting insulin and a GWAS where individuals exhibit impulsivity and did not have any exposure to adversity, again, separately in males and females. So now I want to show you some preliminary results we have from this study. So here are the results from MR from the fasting insulin and no exposure to adversity. So the males are on the left-hand side and the females are on the right-hand side. I ran MR with 4 different methods that were available for 2-sample MR that are listed here in this table. And on the top is the pleiotropy Egger test that I was mentioning earlier. So in green, what I've done is I've highlighted any p-values that are of significance. And what I want to highlight here is that in both males and females, we see that the horizontal pleiotropy test was significant, which basically violates the assumption, and we cannot here be certain that horizontal pleiotropy does not exist. So this essentially means that our results are invalid, no matter what they were. And then we did this again for the yes adversity group, set up the exact same way. And the pleiotropy assumption is violated in males, but not for females. So for males, the results were invalid, no matter what they were. But in females, we can actually look at these results. And we see here that every MR method shows a significant result for causation. So these are all preliminary results, but together so far, what they're showing us is that in females exposed to adversity, there is a causal association between fasting insulin risk and impulsive behavior. And in all other cases, we cannot yet determine the causal association. So very quickly, I just want to go over what I'm currently working on based on these results. So as you saw, the major issue we've had so far with these MR analyses is that our results are invalid in most cases because we can't be certain that the horizontal pleiotropy wasn't present. So to tackle this issue, we're conducting an MR analysis in which you can actually identify SNPs that are contributing to the horizontal pleiotropy, and then you can remove them as outliers from the analysis. So then hopefully after doing that, I will be able to show you some valid results in which I will be able to tell you whether there is a causal relationship between the risk for fasting insulin and impulsivity in the presence or absence of adversity. And that's it. Thank you to everyone here for listening and for everybody who's helped me throughout this project. Thank you so much.
Have you considered looking into kids diagnosed with diabetes?
No, because we wanted to make sure that adversity was the one that was playing a role, and early life adversity wasn't resulting in diabetes. It was just a sensitivity to insulin that was happening from very early on. So we haven't looked into that, but I do wonder if just having diabetes could be considered also an adversity, and that could be affecting things. The postnatal adversity score that we do use encompasses a bunch of different adversities, and some of them do involve metabolism aspects in it. So I wonder if that's supposed to included as part of it, but not specifically with diabetes.
All right, thank you.
Yeah, no problem. Thank you.
Hello, everyone. I will discuss how peripheral microRNA profiles in adolescents can serve as potential biomarkers of risk for depression. As you have just heard from other speakers, that early life adversity is associated with neurobiological alterations that impact the well-being across the lifespan. And here I'm summarizing this briefly with the adverse childhood experiences pyramid. And particularly, I would like to highlight the fact that early life adversity and early life stress are well-recognized risk factors for neuropsychiatric conditions. The top contributor to disability worldwide is in fact the major depressive disorder, which has an onset beginning as early as adolescence. And adolescence, the development is still ongoing, and in fact the prefrontal cortex is undergoing major maturational changes during this time, prefrontal cortex being the executive center. Now, the global pandemic has been hard on, that we have all witnessed, has been hard, particularly on the youth, with many reports suggesting there is a surge of cases for depression. And my research is not just interested in the biological mediators for depression, but also biomarkers that can aid us in predicting those that are the most at risk. As it has been mentioned before, the early adversity experience does not necessarily guarantee that a person goes on to develop neuropsychiatric disorders, but there are individual differences. And the last thing that I would like to mention is the fact that as of now, the screening and diagnosis is primarily done based on self-reported symptoms, which is, of course, understandably a challenge for individuals that are transitioning from childhood and are experiencing undergoing many novel experiences. As some of you may know, the genome is filled with protein-coding genes. However, they make up only 3%, and the majority of them being the non-coding RNAs, which are not just the transcriptional noise. In fact, there are many types of non-coding RNAs. My research is specifically interested in microRNAs, and they're known to target many genes at a time and shape epigenetic control of post-transcriptional functions in some instances in an instantaneous and reversible manner, which of course is of great value for disease prediction and treatment. Now, as I mentioned, microRNAs are 20 nucleotides in length, and they have been implicated in neurodevelopmental processes, including neural stem cell maturation into neurons, neurogenesis, dendritic maturation, and another superpower that microRNAs possess is that they're so small that they can cross the brain-blood barrier. So microRNAs found in the brain can be found in the circulating blood. In the context of adolescence, there's a study looking at microRNA, the global microRNA expression in the prefrontal cortex across the lifespan. So from neonates to older adults are finding, in fact, that adolescence coincides with a shift in global trajectories, where some microRNAs take on the role of being upregulated and some are downregulated, and this pattern is being maintained for the rest of life. Now, in our studies, we have identified a candidate microRNA called miR-218. We have found that it is relevant to the prefrontal cortex development, and we're finding that consistently this microRNA is downregulated in the prefrontal cortex of individuals who were diagnosed with depression and died by suicide. We were also able to replicate this finding with the same direction of change in mice, and in fact finding that the differences in microRNA expression in the brain are also paralleled in blood, thus suggesting that this microRNA can potentially serve as a proxy of the readout of the brain expression. Now, without going into much of the experimental details, we did an experiment in animal models of stress-induced depressive-like behaviors. And tracking miR-218 in blood longitudinally showed that we can actually predict mice that go on to be susceptible to stress-induced depressive-like behaviors. Now, in my studies, I'm particularly interested in profiling global microRNA expression in adolescents. So in the human cohort based on blood samples, which would serve as a readout of brain development and activity, and of course aid in identification, prevention, as well as tracking the status of disease over time. The specific objectives are as follows: to capture developmental microRNA expression and assess potential sex differences. As we have heard, sex differences are quite critical, especially when we're talking about major depressive disorder, which has this bias. Another objective is to assess the global peripheral microRNA profiles that distinguish clinically depressed from non-depressed youth. Investigate how alterations of microRNA are associated with the early life adversity history, and as well as dive into the world of candidate microRNA profiles, so microRNAs that have been already identified to play a role in neurodevelopment, and how these microRNAs measure up with the developmental and behavioral outcomes in a cohort, adolescent cohort. And this ambition would not be possible was it not for these incredible cohorts. So one being GUSTO, which is an abbreviation for Growing Up in Singapore Towards Healthy Outcomes. It's a birth cohort with the goal to understand how conditions in pregnancy in early childhood influence the health and development of women and children. The second being TIGER, which stands for Teen Inflammation Glutamate Emotion Research Project, which is a longitudinal study to investigating inflammatory and glutamatergic pathways that contribute to the recurrence of depression in adolescents. Now, the two cohorts that I will be studying have different types of samples. So TIGER cohort has dried blood spots. So these are actually the easiest way of collecting biosamples. It doesn't require a huge lab to have this, and in fact, dried blood spots are frequently used in disease assessment in newborns. Now, my project comes in also troubleshooting, can we use these dried blood spots to isolate microRNA and measure its expression levels? And the GUSTO cohort, we suggested that plasma be isolated from whole blood in order to capture the free-floating microRNAs that are present, which of course we would like to measure as a readout from the brain expression. And the two cohorts, the big difference is that the TIGER cohort is adolescents that have been specifically recruited based on the diagnosis of depression or the lack of psychiatric history. And Gusto cohort is a birth cohort, so it actually has been followed up longitudinally, and the kids are now in their early adolescence. With that, again, microRNA will be isolated and sequenced, thus having this unbiased transcriptional profiling. The demographic slide here that I would like to point out mostly is the fact that, again, the tiger is based on the depression diagnosis, whereas the Gusto cohort, here we selected individuals based on their scores on the child depression inventory. So this is one of the measures that is trying to identify signs in children that they might be at risk for depression. And so some children have low score, some mid, and some high. And so we selected individuals to have this equal representation between the groups. Now, the results that I'm going to share with you are quite preliminary, so there's a lot still going on. And in fact, so far, I'll just focus on Gusto results. The tiger results are still being analyzed. So the first result that kind of stands out is the fact that we're able to see some sex differences, where some microRNAs, 2 particularly, are passing the false discovery rate threshold and are significantly expressed in the blood between males and females. The second one here, as you have seen in previous presentations, this is the volcano plot. So this is microRNAs that are differentially expressed. This is based on the order of magnitude, the magnitude of change. And these microRNAs are upregulated and these microRNAs are downregulated. As you can see, there are several differentially expressed microRNAs. With the large circles here, I created in order to identify those that are passing the false discovery threshold. So there's 4 that are passing the threshold. However, if you look closely, ooh, that is a messed up slide. My apologies, but the MIR218 that I have talked about in my earlier slides, we're actually seeing the same direction of change in this cohort as we did in our animal studies, so that's quite fascinating. And the second thing is that looking at the literature at some of these microRNAs, MIR375 has actually been identified in a study that was looking at paternal loss and maternal separation in childhood, and they were looking at children as well as adults, and they're finding this upregulation of this microRNA, which is also similar to what I'm seeing. And again, remember, this is a community-based cohort, so quite remarkable. So the 4 microRNAs that I have mentioned that have been that are passing the false discovery rate, here mainly I would just like to show the fact that some microRNAs are are independent of the sex, and some seem to be sex-specific. So here, for visual purposes, I created the 4-group analysis. This is violin plot to show you the distribution of microRNA expression. As you can see, some microRNAs have the same change between the sexes, whereas others have this pool from one sex only. So that's quite interesting as well. And just briefly mentioning that when I annotate these microRNAs, again, the ones that are coming up as significant, there's a way for me to look at literature reports to see what these microRNAs have been reported to be involved in so far. And interestingly, pain comes up as one of the top associations between microRNAs that are expressed between high versus low child depression inventory scores. So, and of course, the association between depression and pain is not novel and is well recorded. And one of the last things I would like to mention is that in Gusto, the children have the child depression inventory scores from different time points. And in fact, we can see children that are scoring high stably, so they have a high score between different time points, or it changes. And so when looking at that stable between unstable high child depression inventory groups, actually quite a few microRNAs come up. So of course, there's a lot to be looked at there. And as I mentioned, this analysis is very much preliminary and a lot more to be done. One of these things includes looking at the multi-group comparison. So looking at high unstable, high stable, and low score interaction, as well as looking how these microRNAs are correlating with continuous measures instead of just the categorical high versus low. Another thing we can do is look at what these microRNAs are targeting. So microRNAs target several genes at a time. So looking what are those targets, and then creating gene networks such as the WGCNA to assess those associations. And finally, this is actually one of the up-and-coming topics in the field, is the fact of integrating molecular, clinical, and perhaps even neuroimaging measures to see if we can predict the risk for depression in these children before the diagnosis is even made. Now, just briefly summarize what I've talked about so far is that early life adversity is a major risk factor for psychiatric pathology. However, not all are affected equally. So there's this great value in identifying those that are most at risk, as they would go on to likely develop more severe clinical depression. And there's, of course, a dire need for objective assessments to diagnose depression, particularly at-risk children and adolescents, as they may not display the depression symptoms until later in life. And as my project has shown, that performing unbiased profiles of microRNAs in blood has identified some potential candidates that can then further be studied for specific biological processes and mechanisms involved in pathogenesis. Now, it's a great honor for me to be here in front of you all, and I would like to thank my advisor and my collaborators who have made this project possible, and thank you for your patience and attention.
Hey, I had a quick question.
Yeah.
So given that microRNAs have to be constantly retranscribed, I wonder what is the molecular link through which this sort of exposure early on in life leads to long-term changes in expression of microRNAs, knowing that they're constantly retranscribed? Are there other epigenetic mechanisms that are leading to that sort of change in gene expression throughout?
I'm not sure I understand your question. So microRNAs are targeting, or so their role is not that they're coding for a specific function, obviously, or specific protein, is that they're targeting these genes that are together in concert, are having biological functions. So I'm not exactly sure what your question is.
Yeah, just because they have to constantly be retranscribed, right? What leads to that sort of long-term consequence of childhood abuse, knowing that they're constantly... So if you look at other epigenetic marks like chromatin configuration, for instance, it makes sense that early life adversity leads to a configuration that is sort of stable.
Mm-hmm.
So I wonder what the mechanism is through which you see stable changes in microRNA expression from an exposure that happened, let's say, 15, 20 years ago.
Mm-hmm. Yeah, so I definitely have to think about this. This is a very good question. But just off the top of my head is the fact that here the basic goal is to kind of capture the temporary window, what is going on right now, and whether using these microRNA levels as they are, whether they can serve as a readout of what's going on in the brain, as well as potentially predicting how they might look like in the future. So, but yeah, so I definitely understand that this is something I have to think about. Thank you.
Thanks so much.
Yeah. Okay, thank you, everyone.
I think Ed wants to say a few words, but before he does, so thanks to our 4 speakers, and thank you all for attending the symposium, and I hope you'll have a great lunch.
Yeah, thanks, Naguib, for organizing a great session, and thanks for all the speakers this morning. Just a couple of comments. So we'll have lunch now. There's a falafel provided for lunch, again, like yesterday, and you can take it outside, enjoy it. The posters should be up. Please come back, take advantage of the posters. One request I've had is that if you are, some people are uncomfortable with having their posters photographed. If you're taking it for your own personal use to recall what was in a poster, just get the permission of the poster presenter first, just as a courtesy. So that's something that I've had a request for. And come back at 2 o'clock. We'll hear from the Tannenbaum Open Science Institute about open science at McGill, followed by the last session this afternoon on cellular and animal models of disease, and then the career workshop. So thanks again and enjoy the lunch.